Thank you very much. I want to thank you guys for inviting me. I think I have had a great day and met with lots of you already, and so I've really enjoyed my time so far. And so I'm going to tell you a little bit about membrane protein modeling and what I do. And so very briefly, I am right in the middle of Manhattan in New York City, and so I'm at the Simons Foundation, and one of the divisions of the Simons Foundation is the Flatiron Institute. And so you've probably seen the Flatiron building, and so right across Fifth Avenue, there's the Simons Foundation is here, and the Flatiron Institute is right next to it. So this is where I'm located, just to put that a little bit into perspective. I'm also associated with NYU, where my boss also has a lab in the biology and computer science department. So this is the outline of my talk. I'm just going to stand over here to, this is the outline of my talk. I want to tell you a little bit about membrane protein modeling, why I'm interested in modeling membrane proteins in the first place, the difficulties that we have in studying them. I want to tell you a little bit about the Rosetta software and the Rosetta MP framework that we've implemented, and then recent developments in Rosetta MP or that are based on Rosetta MP, so a bunch of different modeling problems that we have for protein-protein docking, high-resolution refinement, and domain assembly. And then I want to put this work a little bit into context in into membrane protein modeling or membrane protein structural biology. So let's get started. Why membrane proteins? Or in the first place, actually, since you're a chemical engineering department, a lot of you are studying material science or other different types of aspects. Protein structure is important for drug development and understanding human health and disease. So, proteins are basically the basic machineries that pretty much accomplish anything in the cell. If we know protein structures and we can put that into context of other proteins and also small molecules, we can figure out how small molecules bind in the protein, we can develop better drugs, and we actually get a molecular level understanding of health and disease. And so this is the basic idea for studying protein structure and especially also membrane proteins. So membrane proteins are very important drug targets. Here's just a table of important drugs that target membrane proteins. You've probably heard about most of these. Abilify is an antipsychotic that targets the dopamine receptor. Here's also sales that this drug had in 2015. So you've heard probably also about Gleevec that targets cancers, Herceptin for breast cancer, and you might actually take some of these against allergies or heartburn. So these target membrane proteins, and membrane proteins are also just important in everyday life. Rhodopsin is the membrane protein that is important for your vision. Coffee as well as chocolate both target the adenosine receptor, which is a G protein coupled receptor. The sense of smell is related to olfactory receptors. If you're smoking weed, it targets the cannabinoid receptor. If you eat sugar, it has an impact on the insulin receptor. If you're smoking, that targets the nicotinic acetylcholine receptor. Your heartbeat is accomplished through potassium channels as well as sodium channels. You also have potassium and sodium channels are targeted by chili peppers, or if you get stung by certain spiders or scorpions. And if you feel cold, that is also a sensation that is accomplished through potassium channels, specifically TRPV channels. So there's just like really important in human health and disease. And so that's why I think they're interesting. So at the same time, so they're incredibly important drug targets. So 50 to 60% of drugs currently on the market target membrane proteins. So even more than what we have for soluble proteins. However, if we look at the number of structures in the protein data bank, only 1 to 2% of those structures belong to membrane proteins. So that means they're vastly underrepresented in terms of structure. And so we basically need better methods to study them. The reason why they're so underrepresented is that there's just a lot of problems in studying them and getting their structure. And so there we go. So you run into a whole bunch of problems studying membrane proteins. The first one is going to be overexpression. So you need enough protein to study it in the first place. And so a lot of times what you do is you want to express those proteins in bacteria, but those proteins are typically toxic to the host cell, or in many cases are toxic to the host cell that you're trying to express it in. So you cannot make enough protein. The second difficulty that you're running into is that since it is a membrane protein, it requires a membrane mimetic to keep it properly folded. And so there's various different types of membrane mimetics that are available, and so you have to figure out which of those is particularly well suited for the type of method that you're studying with. And there's also a whole bunch of other problems that come along with it. So once you have tackled those two challenges, you probably want to do structure determination, and you're probably trying crystallization. So a lot of membrane proteins just don't want to crystallize, and people spent years and years and years of trying to crystallize them without success. It might require extensive protein engineering to keep them in a certain stable state. If you can't do crystallization, you probably want to try NMR spectroscopy. However, NMR spectroscopy really works well for small proteins, but is not very well suited for large systems like membrane proteins, because the tumbling time that goes into that, or basically the quality of the data that you get, depends on the size of the protein membrane mimetic complex. So the bigger that complex is, the worse your data looks like. And so the fewer restraints you can get out of it. Another technique that has recently come up, or has been very successful, is cryo-electron microscopy. So currently this works really well for large systems, but also has a size limit towards smaller proteins. And so there's a lot of, a bunch of different methods out there, but each of those methods has certain problems associated with it, and just membrane proteins in general are hard to study by any of these methods. And so these are just a couple of examples that I have. The first one is the beta-2 adrenergic receptor, which is a G-protein coupled receptor, which was determined by Brian Kobilka and Ray Stevens' lab. I think this was published in 2006 or 2007 after about 20 years of effort. And so this was one of the reasons why Brian Kobilka received the Nobel Prize for his work on GPCRs. This structure is an NMR structure. This is diacylglycerol kinase. It's a homotrimer. This was actually determined in one of my grad school advisors' lab, Chuck Sanders' lab, and this took 13 years to determine the structure. The structure down here is an aspartate protease, also a very large crystal structure published in Nature. And if you read the methods section very closely, they required 160,000 crystallization trials to set this up. Now, from a perspective nowadays, 160,000 crystallization trials is like, all right, you just dedicate a robot for a couple of months to do that. This work was actually done by hand, and it took like many, many years to accomplish. And so this is also the reason why a lot of these structures are still published in Nature and Science journals, simply because of the amount of work that goes into it. So it's really extensive. Also, the membrane protein, like the labor or the amount of money that actually goes into determining membrane protein structures is about 10 to 20 times as much as for soluble proteins. And so it's really a lot of work. So if you have succeeded in actually getting a crystal structure or an NMR structure, the quality of the structure that you're getting is typically quite a bit worse. So, and I have an example over here for crystal structures. So this is basically the resolution that you can get. So this is, if you cannot read this, is the 0.5 angstrom resolution where you can see individual atoms. This is 1.4 angstrom, 2.5 angstrom, 3.5 angstrom, and 5 angstrom resolution. So the resolution gets worse from left to right. Soluble proteins are typically in that range. around 1 to 2 angstrom resolution. So this is a high resolution crystal structure. Membrane proteins, if you get a 2.5 angstrom membrane protein crystal structure, you're really lucky because this is one of the highest resolutions you can get so far. Typically, it's more like 3.5, 4 angstrom resolution crystal structures is what you would get. And at these resolutions, you can get an idea of where side chains are, but it's not easy to pinpoint that very clearly. If you study membrane proteins by NMR, the number of restraints that you're getting, so restraints are basically mostly distance restraints or orientation restraints, those restraint sets are typically sparse. And what that means is that on average, you get a sparse restraint set is about 1 restraint per protein residue that you can achieve. Most of soluble proteins, or that you can get, structures of, have rich restraint sets of about 10 restraints per residue. So, and that basically gives you an idea of, the quality of a structure is just much worse with sparse restraints. Another problem that you will have is that the membrane mimetic that you're determining your structure in has an effect or can have an effect on the structure. And this is an example of a modulatory protein for a potassium channel that was determined by NMR spectroscopy in micelles. And so this is what the structure looks like. And so this is basically modeled onto basically what a micelle would look like in that case. And so this is definitely not how this protein would look like in the flat membrane bilayer because the N and the C termini are basically wrapping around the micelle that it's determined in. And so if you really want to understand how membrane proteins function, first of all, you need to have a high quality structure. And this is not necessarily how a protein would look like in your cell or in your body. And so this is another problem that you have to deal with. If you look at membrane protein methods or computational methods, this is from a review that I published 3 years ago. There's actually very few methods available, and those methods are highly specialized and they're not very well integrated. And so, there's methods out there for de novo structure prediction. There's few methods for comparative modeling if you have templates available. There's definitely molecular dynamic simulations that people use a lot, but these simulations require a very long runtime. There's one method available to model beta barrel proteins, and then there was one method available for loop building. So that was basically it. Now, on the other hand, if you look at the Rosetta software, Rosetta has a lot of different modeling applications available. And this is just a relatively small subset of it, whether it is for de novo structure prediction, comparative modeling, protein-protein docking, protein-small ligand docking, loop building, modeling symmetric proteins, protein design, modeling carbohydrates, predicting delta G's of mutation or delta G's of binding. There's just a vast array of methods available that has been developed for soluble proteins. And so, just a really brief introduction into the Rosetta software. I think a lot of you already know David Baker's lab pretty much next door, but I want to give you a little bit of the history of Rosetta. So, David Baker was basically, or the development of Rosetta started in David's lab in about the mid-90s. And the first paper was published for de novo structure prediction in 1997. Later on, there was setup of BOINC. Basically, if you had screensaver time available, you could install Rosetta at home, and you could dedicate your free computational cycles to research where you could basically fold proteins with that. In 2003, a de novo design paper came out, so the protein is called TOP7, and so this was published. Then Rosetta until here was developed in Fortran, and then it was ported into C++. It was basically an automatic translation, machine translation of the code into C++. and we called this Rosetta 2. So this happened about 2005. From the membrane protein perspective, in 2006, low-resolution membrane protein structure prediction became available. 2007, the one for high-resolution membrane protein structure prediction. So this was 10 years after the first algorithm were developed. And then people realized that the current state of the Rosetta software was not maintainable or not extensible very well because it was automatically rewritten into C++. So what they decided to do, they basically decided to start over and start from scratch and write Rosetta into a new object-oriented code base. And so this is what we call Rosetta 3. Currently, so now 10 years later, we're still working with Rosetta 3, and it has grown substantially since then. In 2008, a video game came out that was based on Rosetta, or basically is running Rosetta underneath. It's called Folded. I'm not sure whether you've heard of it. It's actually a lot of fun to play. You can do some real science with it, so I also use it as a teaching tool. In 2009, in Jeff Gray's lab, where I did my first postdoc, PyRosetta came out. So it's basically a Python wrapper around the Rosetta code base. And in 2011, a Rosetta scripts interface came out where you can run Rosetta based on XML scripts. And so these are basically 3 different interfaces that you can run Rosetta in addition to the command line interface that it already has. The Rosetta community has grown quite substantially over the past 20 years. This is a picture from, I think, from last year's RosettaCon. So every year in the summer, we come here to Seattle for, first of all, the developers meeting for a day or 2, and then we drive up to Leavenworth to have our annual RosettaCon for a few days, where we actually talk about recent developments, we talk about the code base, we talk about how we can solve problems, maintain the code, and all the awesome science that's going on with it. So this is right now less than half of the community. So it's been a huge community so far. And so just a couple of numbers or a couple of facts. Development started in 1997. Currently, we have 52 labs worldwide. Most of these labs were either grad students or postdocs that come from David Baker's lab over the years. We have over 400 developers, even though probably about 100 of those are really active developers committing code into the code base. We have about 2.5 million lines of code base. So it's really, it's actually bigger than Facebook. We have over 20,000 licenses to both leading pharmaceutical companies as well as academic labs. The research is featured in over 1,500 publications, and this number is actually more like a lower estimate. We have several different interfaces, and one of the recent successes is ChemE, which is an enzyme to break down gluten in the stomach for treatment of celiac disease, which is currently undergoing FDA trials. So these are just basically a couple numbers. If you want to know very basically how Rosetta works, we're not doing molecular dynamic simulations. This is a Monte Carlo-based approach. So we basically take a protein structure and make random moves in the structure to perturb the structure, and we do this in a Monte Carlo sampling approach. And then the new conformation that we come up with, we evaluate both conformations using a score function that is both physics-based as well as knowledge-based. And so this is the kind of very basic idea of what we're doing here. And so each of those protocols has those 2 components to predict something. And that something is based on whether you're looking at protein-protein docking or de novo structure prediction or homology modeling or anything that you're interested in. So I want to tell you a little bit about the Rosetta MP Framework that we have developed for membrane protein modeling. And so the basic idea was really, when I started my first postdoc, I had the idea, I was actually, during graduate school, I was also doing experiments, and I realized, oh my gosh, studying membrane proteins is really hard. If we could use a software like Rosetta that has all those tools already available, and if we could extend this for membrane protein modeling, we could really have a huge impact. And so this is basically what I came for my postdoc with. So I basically joined a lab at Johns Hopkins to learn how to use Rosetta and how to develop in Rosetta. And basically I wanted to create a framework that sits right in the middle and interfaces with those different applications to extend those for membrane protein modeling. And so this is actually something that I've done. Also, one of the reasons why this needed to be done is the following. Just historically, in 2006 and 2007, there were the de novo structure prediction protocols, or the score functions, were implemented into Rosetta, and this happened right before a major rewrite happened. So all of the, or most of the new frameworks that are available for structure prediction happened or were implemented after that happened. And so this original code doesn't actually interface with most of these frameworks. And so really to easily develop new protocols, we really needed to take that code and rewrite it completely and hook this into most of those frameworks in Rosetta. And so it was really a software engineering approach or requirement. So in here are just a couple of design standards that we set out to make this available, or to make this happen. So first of all, of course, we wanted to create new science. So we wanted to model large proteins. Rosetta was not set up for that, or still is not necessarily set up very well for this. We wanted to model multi-chain membrane proteins. We wanted to implement new protocols, and we also wanted to think about, well, what do we want to model in the future? What do we want to model 10 years from now? We wanted to make sure that the code is general, that it is maintainable, and that it is extensible. From a code design perspective, we wanted to make sure that this code really hooks into the current setup in Rosetta and interfaces with a lot of existing frameworks in there. And then thirdly, we wanted to make sure that the user interface is user-friendly and that it can be used in the interfaces that are available for use, which are basically command line, PyRosetta, Rosetta scripts. And so these were basically the three requirements that we had for this new framework. And so the work that we did took about 2 years to complete, really going from coming up with a code design, how certain classes would interact in Rosetta, implementing some of that, realizing that it didn't work, and basically going iteration by iteration to figure out a best way of how to do this. And so not to go into too much detail, but basically what this is showing is that we've implemented that framework in a way that it extends current Rosetta, or the current setup in Rosetta. So basically every, what we call protocol, or every tool that we're looking at has three main components. And the first one is the pose, which is basically the biomolecule that you're modeling. Most of the time this is going to be your protein. Then we have movers available to make a certain change in that structure, and then a score function to evaluate whether that change was a good change or a bad change. And so we extended this for a membrane protein, so we implemented membrane movers to sample in a smarter way. We implemented membrane scores for the score function, and then we have a certain membrane-related information that we're tagging onto that biomolecule object to make sure that we're modeling a membrane protein. One of the requirements was we've actually added a virtual residue to be able to visualize membrane coordinates. And so this was something that the previous application could not do. So you would see your protein, but you wouldn't actually know how it would be embedded in the membrane. And so this was a really major drawback of the old code. And so now we have fixed this. We actually hooked this into a software that we're using for visualization. It's called PyMol. And so we can actually allow real-time visualization of the protein embedded in the membrane for debugging, for making movies, and developing protocols. So we also tested for proof of concept, or we implemented for proof of concept application to show that we can combine that Rosetta MP framework with existing protocols. One of them is high resolution refinement. We call this MP Relax. One of them was modeling symmetric assemblies, because a lot of membrane proteins are symmetric. We call this MP SimDock. Prediction of delta G's of mutation. Basically correlating experimental DDGs of mutation with Rosetta predicted DDGs of mutation. I'm fully aware that this correlation looks very terrible from for an untrained eye, but it's a really, really difficult modeling problem. The fourth application is actually protein-protein docking, which I'm going to go into next. So protein-protein docking. Perfect. Okay. So let's talk a little bit about local protein-protein docking, and just to go back really quickly. So you basically take the protein-protein docking application that was available in Rosetta, and you combine that with the Rosetta MP framework. And so in this case, this is local protein-protein docking, which means that you know where the interface is, you know how the two proteins interact with each other, but you're refining that interface. And so this was really the easiest part of that. And so the next video, I'm actually going to show how we've set this up. So the first thing that you're going to see is the membrane protein is added, the membrane residue is added to embed that protein in the membrane. You saw that the side chains kind of disappeared. We got rid of the side chains. We do a centroid-based or low-resolution docking trials until we find a reasonable conformation. And then once we have found this, we actually add the side chains back on, and then it goes into high-resolution refinement, which I'm not showing in this video. So this is just very basic sticking Rosetta MP together with protein-protein docking. So however, the problem is also that a lot of times you might know from an experimental point of view that two proteins interact, but you actually have no idea how they interact. And so especially also since membrane proteins are important signaling molecules, let's say you have a signal coming from the outside to the cell, they create some conformational change that is somehow transmitted through the membrane, and then another conformational change or molecules come along to bind to actually create a signaling cascade inside the cell. And so really to understand that biology, we need to understand how proteins come together in the membrane. And so another thing is also membrane protein complexes are far more difficult to study than individual membrane proteins. So just from an experimental perspective, it would be much easier if we could come up with a model and say, this is how this might look like. Please test this based on mutational data or other types of experiments, rather than crystallizing the complex together. And so this is actually what we call a global docking problem. And for soluble proteins, that global docking problem looks like the following. You have two proteins floating around in solution, and each of them have 3 degrees of rotation and transformation or translation, and you don't know how they come together. So the conformational search space is enormous. However, if you study membrane proteins, the problem becomes actually quite a bit smaller. First of all, you're sitting in a 2-dimensional membrane. Secondly, your proteins can only come together either in a parallel or anti-parallel way. And so this really cuts down on the conformational search space quite a bit. And so the question is, maybe for membrane, while the global docking problem for soluble proteins is currently still unsolved, maybe we can make this happen for membrane proteins if we develop a protocol that can efficiently sample. And so what I set out to do is I implemented a protocol for global docking of membrane proteins, but also flexible backbone docking, because proteins move they're not rigid molecules. And so this is a very basic overview of how this protein looks like. We add the virtual membrane residue, we move the two docking partners apart, we do a high resolution refinement, we do a couple of docking trials, which is based on 4 movers, and those 4 movers are spin movers, translate movers, tilt movers, and flip movers. And so we do this a couple times, then we do another high resolution refinement before we score and output the model. And what this might actually look like in real time is something like this. So you have the native structure, or the NMR structure, in gray, and in blue is the model that they're actually building. You saw that the side chains were, the side chains in the backbone were refining at the beginning, and then you do global docking trials. So these are the movements that you're making. And then if you found a reasonable interface, you do another high resolution refinement step to really refine that interface. And so this is really the basic idea of how this might look like. So another thing that needs to be taken into account here, or that we've noticed from the data that we're getting, that increasing flexibility, and that flexibility would mean backbone flexibility as well as sidechain flexibility, changes or leads to a different ruggedness in the energy landscape that you have to deal with. And so basically over here we have a fixed backbone and a fixed sidechain, and what we're looking at is at the interface score versus the root mean square deviation of what this might look like. And so what you actually want to see is a funnel like this, where the lowest scoring model is close to what we call the native, close to an RMSD of 0, because that means that we can identify a good model by a low score only. However, if you include more and more flexibility, and over here, this means we have flexible side chains, that depth of the energy well is not given, or is not as deep anymore. And so the more flexibility you actually include, the more broad your energy landscape looks like. And so these are definitely things that still need to be addressed in both sampling as well as scoring to really make this protocol a really well-working state-of-the-art protocol. In terms of runtimes, because this is also something we don't want to have to run this for 6 months to get some data, I develop the protocols always looking for, quality of the data that I'm getting out, but also runtimes. And so over here you have a number of residues. You can actually dock 1,200 residues within 10 minutes, which is, from an MD simulation perspective, is really, you cannot possibly do this with MD simulations. And so in this protocol, there's definitely a lot of things that still need to be done to improve the score function for protein-protein docking, look at statistics for membrane-protein interfaces and whether we can actually derive some knowledge-based potentials to add to that to be able to identify good scoring models, benchmark this protocol, and then of course there's lots of application projects that I have access to from collaborators who want to dock membrane proteins together. The next step, or the next protocol that I want to talk about, is high resolution refinement. And this is really an incredibly essential protocol because it's used at the end of pretty much every modeling protocol out there. It's also used to create starting models for protein-protein docking, for protein ligand docking, for protein design. It's also used to model ensembles. And so, especially for membrane proteins, this is incredibly important simply because the quality of the data that we get out from crystal structures or cryo-EM structures is very low resolution, and we need a good refinement protocol to refine these to high resolution structures. And the same thing is also for modeling ensembles. So this protocol is really incredibly important. However, the way the current refinement protocol is implemented, since historically Rosetta was developed for small soluble proteins, which is exactly what membrane proteins are not. They're very large systems typically. And so we somehow have to deal with this. And so the high resolution refinement protocol which was implemented, which was called fast relax, is anything but fast, especially for large proteins. So typically soluble proteins are in the range, 100, 200 residues. And so when you refine such a small protein, you can get, good quality models within a few minutes, 10, 15 minutes. However, if you look at a membrane protein where you can easily have 2,000 residues, it would take you a single day to build a single model. And this is just not realistic if you need to have those applications available, especially also as part of other applications that you're building around it. So this is simply not feasible. And so the reason why this is actually the case is how the protocol is set up. It ramps full atom repulsive scores up in the modeling protocol. Then it does a sidechain repacking, minimization. It does that, 5 times. And so that entire setup is very inefficient simply because the sidechain repacking is inefficient simply because it's done over the entire protein at once. And so imagine that you have a sidechain that clashes with another sidechain. And if you repack your sidechains at once, what is happening a lot of times is that suddenly it clashes with a different sidechain and you actually haven't achieved anything. And so what I've actually developed is a protocol that can deal with that much more efficiently by doing the repacking from the inside out. So basically, I define shells from the center of mass of the protein where I repack residues from the inside out. That means the side chains can always move. They will not clash with each other, or at least not as often. And so I've implemented that protocol. I've called this the range relax algorithm, which was basically designed for speed, but I'm also going to show you some results. I've implemented this in a way that we can use this for both soluble proteins as well as for membrane proteins. And then this is the very basic protocol of how this might look like. So I repack shells and I do minimization, do this 3 times before I output the model. So these are some results. So the original results that you've seen for fast relax are in red. And so this is a relatively small benchmark set of a few membrane proteins. The largest of this was the photosystem 2, which has over 5,200 residues. So it's a huge membrane protein. And so we can now refine this within about 5 hours. Whereas previously, these 3 guys, over 2,500 residues, with this protocol in red, they wouldn't even run. You couldn't even tackle those systems previously. And here are some more detailed results. So these are PDB IDs for those structures, number of residues, and here is the speed improvement. And so this would be anywhere between, or on average, you would get the 20 times speed improvement, but up to 60 times speed improvement for this particular small benchmark set. I also want to show you what this refinement protocol, or what some of the results of the refinement protocol, if you look at crystal or NMR structures. If you just map the clashes that you get from those structures from the PDB onto the structure, everything in gray means all of those residues are not clashing. Slight clashes are in yellow. Intermediate clashes in orange, and large clashes or really bad clashes are in red. And so you will see that actually every single protein in the PDB has some kind of clashes somewhere. And so if we want to resolve this with the original protocol, we resolve a lot of these clashes, but at the same time there's also still a lot of clashes that remain, but we cannot tackle those really large systems. So this is ordered by size. So this is the smallest one, number of residues. These are the largest systems. We cannot tackle them with the original protocol, and the runtimes for these are actually enormous. And so now if we run the new fast relax protocol, we actually resolve a lot more clashes than previously. We can tackle those really large systems, and we can do so in a much shorter runtime. And so these are really encouraging results for this type of protocol. The last protocol that I want to mention is a domain assembly protocol. And this is, so the basic idea is a lot of times what's happening in the wet lab or when people study membrane proteins experimentally, they realize, oh, this is my construct, and here is my membrane domain. I don't want to deal with it, so I just cut it out, and I study the rest of my protein in isolation. And so what you will find is you will find a lot of times in the protein data bank, you will find bits and pieces or individual domains of those proteins. Sometimes, or you will find that there is a membrane protein or the membrane domain is available as a structure. Sometimes you can model this from homology modeling or de novo modeling if you have, let's say, coevolution restraints available. So you have those bits and pieces, but if you really want to understand protein structure and function and how, let's say, a signal is transmitted through the membrane, you cannot do that if you only look at bits and pieces. And so the idea is that it would be really neat if we had a modeling protocol available that can take those bits and pieces and stitch them together efficiently to be able to come up with models that could be tested in the wet lab. And so this was the idea. So previously, an algorithm like this did not exist. What happened most of the time is that people who are interested in a particular protein system would go to a computational biologist and would say, please stitch this together for me, or please give me a model of how this might look like. And the computationalist would spend weeks or months in actually doing this one by one. So there was no automated protocol available that could handle those cases. All right, and so the way this works is the following. So you basically, your inputs are structures or models of the individual domains, the membrane domain, extracellular, intracellular domain. You have the sequence available of the full-length construct. You start out with the structure in the membrane. You add residues towards the C-terminus. You add the C-terminal domain. You add residues towards the N-terminus, like the linker regions, and you add the N-terminal domain. Then you do fragment insertion in those linkers. You do backbone perturbation and side chain repacking in those linkers. And then you could do, or you're doing energy minimization of those linkers, and you could do an entire high-resolution refinement step, which is optional at the very end. So it's actually, the idea is incredibly simple, but since such a modeling protocol is not available at all, it's like you have to start somewhere. And so if you look into the protein data bank, there's actually relatively few structures available that have a membrane domain and an extracellular domain together. And so we took 4 structures from the PDB, and we took out those linker residues in orange or in red, and then we put that into the protocol and reassembled the structure. So in gray, you will see the crystal structure, and you will see the top RMSD model in blue with the linker region in yellow and red. So the native linkers are in yellow and the model linkers are in red. And so we see that in all cases, we can actually recapitulate native orientations of those different domains with respect to each other. But at the same time, while this is the first step, it would also be really nice to have experimental data to really identify good confirmations. So in terms of runtime, this protocol also runs relatively quickly. So this is over sequence length, so size of the protein. If you have, 700 residue protein, without refinement, you can create models within 5 minutes. If you do refinement on top of that, you can create models within, let's say, for 35 minutes. And so the runtime depends on a couple of factors: linker length, number of residues in the soluble domains, and then, of course, number of residues of the full-length construct. Yeah. I'm currently in the process of extending this for dimers. So the idea is I want to model, for instance, like metaprotropic glutamate receptors. And so there's transmembrane domain structures available, as well as cysteine-rich domain, ligand binding domain structures. And so I want to be able to model those as dimers. Currently, there's an automated sequence alignment, or there didn't used to be an automated sequence alignment method in there, but I've implemented this. I've implemented the loop modeling that's happening within that protocol. And so I need to finish this up and publish this. So I just want to put all of that work a little bit into context of how membrane protein structural biology evolved, or even in general. So this is the growth of the number of protein structures in the protein data bank. The first structure came out in 1976, and now we have about 140,000 structures available. Rosetta started development, or the Rosetta Commons were founded in the mid-90s. So when you had a reasonable number of structures available. So this happened 20 years ago. And this is how this graph looks like for membrane proteins. So the first structure of membrane protein came out 1985, about 10 years later. Look at the counts. Granted, this is the redundant, so this is the number of structures in the PDB, whereas this is the number of non-redundant membrane protein structures. But this is still a lot smaller. Nowadays, we have about 750 non-redundant membrane protein structures available. So we know very little about them. And so this is when I did my undergrad in physics, then I did my PhD over here. The number of methods that were available during that time was very, very little. The number of structures that were available when I started my PhD was about 100 or even less than that. And so now we have a lot more structures available, even though this is nothing compared to what we know about soluble proteins. And so really, while a few years ago we had few methods available for membrane protein modeling, really using Rosetta and implementing such a general framework has already had a huge impact in the number of protocols available, because we've worked on high-resolution refinement, protein-protein docking, symmetric proteins, predicting delta-delta Gs of mutation. And so I'm currently also in the process of tackling other modeling challenges for de novo structure prediction, protein design, modeling beta barrels, and also protein small molecule ligand docking, and all of those in the membrane protein context. And so, and if you look into the future, there's a lot to do. Membranes are incredibly complex. So this is kind of a very simplified picture of what membranes look like. Membranes are asymmetric, so they have an inner leaflet and an outer leaflet that have a different lipid composition between the two of them. They have not just a single type of lipid incorporated. There's, I think, over 600 different types of lipids known currently. So the cell makes a vast variety of lipids. So we want to be able to model that. How do we deal with bending? How do we deal with that the membrane is not just a fixed slab that is just flat? How do we deal with membrane fission or fusion, where two membranes either come together and fuse, or where they blob off and the vesicle floats around in the cell? How do we handle membrane-associated proteins that are sitting on top of the membrane? How do we deal with pore-forming proteins? There's just so many questions out there that need to be addressed and that require different types of modeling protocols, different types of energy functions, and certainly also specific types of sampling protocols to be able to model that efficiently. And so this is something that I'm incredibly excited about. I mean, I've spent my last 6 years of doing this and developing this, And I'm just super excited about all of this new fancy stuff that's going on there. So with that, I want to also thank a lot of people who I've worked with or for over the years. So this is actually my graduate school advisor when I was at Vanderbilt. His lab is actually heavily using the methods that I'm developing, which is really nice to see. This is Jeff Gray's lab where I did my first postdoc. Rebecca, she was an undergrad at the time, and she really helped me. We both of us together implemented that Rosetta MP Framework that pretty much all of my work now is based on. This is my current advisor, Rich Bonneau at the Simons Foundation, and then of course a lot of collaborators both inside and outside the Rosetta Commons. This is definitely not something that I've done somewhere by myself. There have been a lot of people have been involved in that, both with ideas, data, and collaborations. And so I'm happy to I'm happy to answer any questions that you might have. Thank you.
Great. Are there any questions?
I actually have a couple of questions.
Mm-hmm.
And you came back to it, the complexity of the lipid landscape.
Yeah.
And the asymmetry that you have. So a lot of these structures are obtained in Unix. These
Yeah.
have a different lipid composition than native membrane
Yes.
composition.
That's
So
one of the problems.
Right. But you can have also nucleic acid
Mm-hmm.
and very, very complex type of environment. So how do you address the lipids in your calculation?
Currently, we have an implicit potential that doesn't even deal with specific lipids at all. And this was actually something that I've also talked to a bunch of the Rosetta Commons membrane protein PIs. Currently, it doesn't even necessarily make sense to put in specific lipids into Rosetta, simply because then you could just run MD simulations. So currently, we're really using more like a mean field approach that takes the statistics from the PDB and uses the knowledge-based potential based on that, and uses that as a potential. It's incredibly crude. It's definitely utterly wrong, but it's a starting point that we can deal with, simply because, I mean, as you mentioned, like having those membrane protein structures available, the detergents or lipids that are used are completely different than what is actually happening in a native membrane. But that's not anything that we can address anytime soon. And so unless you really have structures in native membranes, which I don't think there actually are.
Part of the reason why I was bringing this up is because lipid fluidity should actually be a constraint on when you're doing your movers.
Yeah.
You might
Yeah.
be able to
Yeah.
do something like this. So if you build, maybe some type of fluidity within
Mhm. 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 Mhm.
you might
Yeah.
be able to actually calculate faster. And the second question
Mhm.
that I had is your mover for your
Global
membrane
docking?
segment,
Yeah.
interaction.
Uh-huh.
You're doing a fluid calculation.
Yeah, and that can be turned off and turned off if you know. I mean, of course, like in some cases, you know exactly where the outside and the inside is,
Yeah.
and sometimes you don't know that.
Because I was going to say you can save yourself a lot of time.
Oh yeah, no, totally, yeah.
So,
Yeah, no, totally. Any more questions?
I got a question.
Sure.
So let's say that somebody discovers a new protein membrane from scratch, like nothing anybody has seen before.
Membrane protein, yeah. Uh-huh.
How do you start the process from scratch? What is the whole process to determine the structure, even from determining how the amino acids are connected with each other and everything?
I mean, like, you know the sequence from sequencing information, so that's your starting point. You're talking about so you said there is no structure available at this point, just like you have the sequence and that's it, right?
Yeah.
So what I would do is the first thing is secondary structure prediction, so to figure out where the helices and the strands are, to figure out where the membrane domain is, and that's membrane span prediction. This is actually something that I worked on in graduate school. So there's methods available for that, which mainly rely on artificial intelligence techniques or machine learning techniques. And so once you know that, then it's a question of assembling those secondary structure elements into a three-dimensional structure. And that's what's called de novo structure prediction. So if you have no experimental data available to help with that, the first thing that I would do is a multiple sequence alignment to identify co-evolving residues. And so this is something that has been shown to work reasonably well if you have what is called a deep sequence alignment. If you have many sequences available that look very similar, and the idea is if you have two residues in space that are close to each other, if you mutate one of them, the other one probably has to mutate as well to counteract for that change. And so this has been shown to work reasonably well in some cases. That's something that definitely I would try first. And so then you can incorporate what I would call smart sampling algorithms to be able to deal with that, to be able to deal with the conformational complexity of the landscape that you're trying to search. So from a computational perspective, that's something that is somewhat doable nowadays. 5 years ago, this was much more difficult or impossible because simply of how the way the math was set up or what they discovered of how to treat evolutionary constraints was just very different. So I'm actually currently working on a protocol like that to be able to do this. From a structure determination perspective, yeah, it's the whole pipeline. The sequence, you need to figure out how to express your protein. You need to figure out in what type of system you can express them, what type of membrane mimetic you want to use, what makes sense, where can you get the best data is one of actually another huge problem because the structures that are out there are not necessarily structures in the best membrane mimetic or in a sense that it keeps the structure in the most native-like membrane environment, but rather where can you get the best data from? And so, yeah, and this can take, so you're going to be busy for a whole bunch of years if you're up to that point, and then you get to crystallography or NMR while you're trying to figure out which other proteins are interacting with and the whole spiel. So yeah, that's basically the idea from both a computational perspective as well as experimental perspective.
Thank you.
You're welcome.
And one last question, Michael?
Seems like one of the biggest issues is just the inability of instrumentalists to determine these structures.
Yeah, it's really, really hard, yeah.
If there's anything that you can gain from the knowledge that you acquire from the computations and modeling that you do that can potentially help advance the technology?
Oh, absolutely, absolutely. So the basic idea is if I can model an interface, if I can model a structure, if I can model where a small molecule ligand binds, I can give that to the experimentalist and say, hey, I think this is what's happening. Or I can say, if the modeling doesn't really pin down one certain conformation, I can say, hey, I have these 3 conformations that are very different. You can design experiments to test these. You can design experiments to say, oh, this conformation is definitely wrong, but this will save you time in doing experiments because you don't necessarily need to start from scratch and need to explore everything. And so the typical pipeline for this is really experimentalists come to me or to us modelers and say, hey, can you help me? We need a model to come up with a hypothesis of how the protein looks like or how it might function. We model this with whatever modeling protocol it requires, give that back to the experimentalist and say, hey, I would like you to run these and these experiments in this priority. They go do these experiments and then it goes back. It's an iterative approach. So it's not just like, oh, we give them the models and then they're happy. No, typically those collaborations are really long-term collaborations because experiments take a really long time. The modeling is also is not trivial at all. So these are really, yeah, take time. Which, sorry, at the same time also feeds back into improving the methods. So this is actually incredibly important for us to see, yeah, this is how we can make this method better, or our results are completely different than what the experimentalists are getting. So that's bad. That really tells us how we can improve the method. So, sorry.
Okay, let's thank our speaker.